Hey, we've all had those days. An occasional fall, trip, or injury happens once in a while. Some may be more embarrassing than others. When this happens, you usually turn towards at-home treatments to help, whether that's ice or heat, plenty of rest, or, of course, medicine. In the case of medicine, a common at-home treatment is ibuprofen. Ibuprofen is classified as a nonsteroidal anti-inflammatory drug, or NASID for short. It is used as a common treatment for inflammation caused by headaches, toothaches, back pain, or even just minor injuries. When you're in pain, NASIDs lower the level of prostaglandins in your body. Prostaglandins are your body's response to inflammation and injury, indicating that there's something wrong. As a result, taking ibuprofen or other medicines containing ibuprofen, such as Advil, Motrin, or Nurofen, helps to decrease swelling and pain. So that's a general basis on how ibuprofen works, but what if there's more to it than that? What if we can take this in a different direction? In general, the overall success of a drug depends on its ability to successfully diffuse into your bloodstream so that it can reach its target, injury, or source of pain location, whether it's taken as a pill, gel capsule, or liquid. This is where transport phenomena comes into play. In simple terms, transport phenomena is the study of how a physical property, whether that be a particle or a moving object, is transferred from one point to another. The diffusion process starts when ibuprofen is taken orally and then travels through the digestive system until it reaches the bloodstream. Once the ibuprofen circulates through the bloodstream, it will reach the target location of where the pain or inflammation is located, decreasing the level of prostaglandins present. For simplification purposes, the remainder of transport phenomena applications will only be focused in the small intestine, as demonstrated later in this video by a COMSOL simulation. But why wouldn't we want to focus on the whole diffusion process from start to finish? The small intestine is the main site where almost all nutrients and minerals are digested. In order for ibuprofen to be effective at its target location, the entirety of the drug must disintegrate in the small intestine and bypass the acidic environment of the stomach. The small intestine is more desirable due to a larger surface area for diffusion, which is aided by permeable membranes. These membranes are lipid-soluble, which conveniently enables lipid-soluble drugs like our ibuprofen to be absorbed, and this membrane-drug interaction will be discussed later. A desirable environment for diffusion is essential for effective drug delivery. Besides just having desirable reaction conditions, what else impacts the rate of diffusion and overall the transport phenomena of the diffusion of ibuprofen? Actually, there are many factors, including but not limited to a person's age, weight, gender, what time they took the medication, their level of physical activity, level of stress, the content and pH of their stomach, and other medications they take. Another user-independent factor is the nature of the ibuprofen taken. In fact, the company that produces Advil, GSK, has different versions available in various delivery methods. The most generic form available, which many of you may recognize, are the burnt red color tablets, which are available almost anywhere. However, there are other versions of ibuprofen available that come in caplets, liquid-filled capsules, better known as liquid gels, and dual-action medications. These ibuprofen alternatives vary slightly in their absorption processes due to their unique diffusion phenomena. With that information, there's a lot of variability and factors that play into the overall success of ibuprofen and other similar drugs. Scientists and engineers are still studying the exact science of medicine diffusion for more standardized information. Unfortunately, we can't Google how much of a 200 milligram ibuprofen liquid gel exists in the digestive system, specifically the small intestine, of a 25-year-old male after 30 minutes of absorption, or a similar question like that, and expect to get concrete and accurate information. So using our knowledge previously discussed, we can create a generalized model where we want to estimate the diffusion coefficient of the ibuprofen. By doing this, we are able to describe how the diffusion process works with respect to time. As shown in the diagram, ibuprofen diffuses from the water-like intestinal fluid across a membrane and into the flowing capillary to be carried away to the rest of the body. This interior membrane is made up of epithelial cells and specifically enterocytes, which are responsible for absorption of chemical species into the blood. The main goal of the simulation was to mimic the biological system as shown in this diagram. By computing the concentration gradient of ibuprofen, we were able to validate the time it takes for the drug to pass through the body. The simulation uses COMSOL multiphysics 2D axiometric modeling with a fully developed laminar flow and transport of diluted species physics. The driving forces for the ibuprofen transport are attributed to diffusion by Fick's law. This means that the rate of diffusion is proportional to the concentration gradient of the ibuprofen, where ibuprofen is diffusing across a membrane of high concentration from the intestinal fluid to an area of low concentration in the blood. Using our current knowledge, we assume that whatever form of ibuprofen is taken, whether it be a tablet, capsule, or liquid gel, it will be fully broken down at the start of our model. We assume that the temperature across the entire system would be that of the average temperature within the body. To simplify the system for modeling, a cylinder was used to represent the small intestine, with a layer for the intestinal fluid, enterocyte membrane, and capillaries. The control volume we used represents a 10 centimeter section of the small intestine, as diffusing across a membrane over the entire length of the roughly 22-foot small intestine can accurately be simplified to diffusion in one spot. As the diameter of the small intestine is approximately 2.5 centimeters, the combined radius of the air and water modeled is 1.25 centimeters. According to a similar study, taking our 10 centimeter system as one intestinal compartment, it would contain around 13 milliliters of fluid over the course of the dissolving medicine. From there, the height of the intestinal fluid can be determined with the following equation. This measurement was then used as the thickness of the intestinal fluid layer, and the remainder of the radius was modeled as air to account for the fact that the small intestine is not completely filled during digestion. Don't worry, there's only a few more important values that I have to mention. We're almost there. The membrane width was set to the diameter of an enterocyte, the width of the capillary layer was set to the diameter of the capillaries, and the velocity of the laminar layer was set to the average speed of blood through capillaries. The material for the capillary layer was set to the density and viscosity of the blood respectively, and the initial pressure was set to the blood pressure in the capillaries. In order for the concentration of ibuprofen to leave the system, the outlet barrier of the capillary was set to the external convection with a diffusion coefficient equaling the average velocity to represent the ibuprofen being transferred into the body through the blood. Now, that isn't even the best part. With the model described, the Navier-Stokes equation is used to model the laminar flow within the capillary. The Navier-Stokes equation is the differential form of Newton's second law of motion for a control volume. In this case, the control volume contains the ibuprofen molecule and drug diffusion interaction in the capillary tube. Using this equation along with Fick's law of diffusion, COMSOL is able to produce a model highlighting the flow of the capillary, which is fully developed for simplification. For a 200 milligram tablet of ibuprofen, the concentration in 13 milliliters of intestinal fluid can be determined using the molecular weight of ibuprofen with the following calculation. Then, we were able to use an initial guess for the diffusion coefficient at around 3.4 times 10 to the negative 11th meters squared per second, which is a diffusion coefficient found for ibuprofen and silicone oil. Since the cell membrane is a lipid membrane, we hypothesized that it would be a relatively accurate representation of the diffusion coefficient of ibuprofen across the lipid bilayer of the enterocyte cell. The COMSOL model simulates the concentration profile over the first 4 hours, showing the decrease in ibuprofen concentration over time as it leaves through the capillaries. The half-life of the 200 milligram ibuprofen occurs around 2 hours. Using this fact in our COMSOL simulation, a more accurate diffusion coefficient can be calculated as 9.5 times 10 to the negative 11th meters squared per second. This was found by dividing the concentration gradient into small sections of a control area, and then summing values that equal R values, and then multiplying the circumference at that R value by the control area. Finally, by summing these values and multiplying by the total molecular weight of ibuprofen gives the remaining mass of the system. Lastly, to further analyze this topic, we wanted to study the pH of the small intestine and ibuprofen's pKa, as they directly correspond to each other and relate to the drug's ability to cross the membrane. In our experiment, a weak base was chosen to model the conditions of the small intestine. This time lapse shows a 200 milligram tablet of ibuprofen dissolving in a 5% weight potassium phosphate dibasic solution. These basic conditions model the environment of the small intestine. The red enteric coating is the first to dissolve. The coating protects the drug while it passes through the stomach until it reaches the small intestine. Once the coating is gone, the inside of the tablet begins to diffuse radially. In the small intestine, this process will be aided by an imposed velocity from digestive processes. Since there is no velocity here and only a small amount of liquid, the drug is broken down but cannot dissolve completely. So with all this information, you're probably wondering, what good is this to me? I'll just take ibuprofen when I need it. Well, yes, as you should, but hopefully it'll make you think about all the connections that drug diffusion has to transport phenomena the next time you're at your local pharmacy.